I am Dr. W. B. Gundule from Kamala Nehru Mahavidyalaya, Nagpur. Today we are going to discuss the protein and what are the classification of protein. That is the different types of, depending upon the properties, we can classify the protein into different ways. And after that also we are going to discuss the primary structure and secondary structure of protein, and then what is meant by denaturation of protein. So the detailed aspect about the protein we are going to discuss in today's presentation. So today's topic is the protein, and here in protein, first what is meant by protein that we are going to see here. As we know, protein is the Greek word obtained from the word proteos, meaning is first. And these protein are extremely complex natural polymer and are first among the organic compound essential for the growth and maintenance of the life. They are present almost in all living cell, and it is found in almost every part of the plant and animal. In human being, they are the main constituent of muscles, skin, hair, nail, tendon, and arteries, and the connective tissues. And we can say that like cell, it is made up of thousands of different proteins. The protein present in the different part of the plants and animal, and even the protein present in different tissues of particular part of the plant and animals from one another in the composition and biological functions. All proteins contain the same element like carbon, hydrogen, oxygen, nitrogen, and sulfur. Some of these may also contain phosphorus, iodine, and traces of metal such as iron, copper, zinc, and manganese. So these are the protein, and as we know that plant alone can synthesize protein from CO2, water, and inorganic nitrogenous compound, but animal mainly depends upon plant for their protein requirement. Chemically, proteins are essentially polypeptide having molecular mass greater than 10,000. So this is about protein. So protein is very essential for the growth of the plant and present in the plant and animal, and protein can be classified into different parts depending upon the number of weights. So protein are classified in number of different weights, and the two important widely used systems of classification are given here. And the first is the classification by chemical composition. So on the basis of this chemical composition, we can classify the protein into three different types. One is a simple protein, second is conjugated protein, third is a derived protein. So on this part with example we are discussing here, and first part is the simple protein. So we can say that simple protein are those which are made up of chain of amino acid unit only. So simple protein contains amino acid unit only and joined by amide linkage. It is on hydrolysis they give mixture of amino acid, and therefore we can say that they are classified on the basis of decreasing solubility, and these simple proteins are further classified into four, that is albumin, globulin, glutenin, histone are the different depending upon the solubility, and therefore they are further classified simple protein into further four. So this first is the albumin. It is soluble in water, acid, alkali, and it is coagulated by action of heat. Example of these albumins are egg albumin, serum albumin, and lactalbumin are the examples of albumin. And second is a globulin. So here globulin, it is insoluble in water but is soluble in dilute solution of salt, inorganic acid, and alkalis. It is coagulated by heat and precipitated by half saturation with ammonium sulfate. An example of such type of globulin is the examples are tissue globulin, serum globulin, vegetable globulins are the examples. And third is a glutenin, and in case it is this glutenin is insoluble in water and dilute salt solution but soluble in dilute acid and alkalis. It is coagulated by heat. Examples are glutenin in wheat, coregenin in the rice are the examples of glutenin. Fourth is a histone. It is soluble in water but insoluble in dilute ammonia. These are not coagulated by heat and hydrolyzed pepsin and trypsin. Examples are hemoglobin, nucleic acid, or histones. Example of this histone. So these are the four simple proteins. And second type is a conjugated protein. So what is now conjugated protein? These molecules are made up of simple protein united covalently and non-covalently with non-protein factor. And non-protein factor is known as prostatic group, also known as cofactor. So it is further depending upon the chemical nature of the prostatic group. These are conjugated protein are further classified into different types, and these are examples are glycoprotein, nucleoprotein, phosphoprotein, chromoprotein, hypoprotein are the different types. And so further they are classified depending upon the chemical nature of the prostatic group present in the protein. And first is the glycoprotein. Example is mucin in saliva, prostatic group sugar, that is carbohydrate. Second is a nucleoprotein. In this case DNA, example is DNA, prostatic group is a nucleic acid. Third is a phosphoprotein. In case of this, example is casein in milk, and prostatic group is a phosphoric acid. Fourth is a chromoprotein. Example is hemoglobin in red blood cell, and prostatic group is colored iron pigment. Fifth is a hyperprotein, and example is protein found in the brain, nerve tissues and blood plasma. Prostatic group is a cholesterol, ester and phospholipids. So these are the different types of conjugated protein depending upon the chemical nature and of the prostatic group present in the protein. And third is a derived protein. And so derived protein obtained by the action of enzyme, chemical agents or physical agents such as heat upon higher protein. The intermediate products are further classified as proteases, peptones, polypeptide, like that. The coagulated protein formed by the action of heat, alcohol, salt of heavy metal upon protein are also example of derived protein. So these are the three different types of the simple protein, conjugated protein and the derived protein. Three are the depending upon the this first classification, and this classification based on the chemical composition. And here, so based on the chemical composition, there are three different types: simple, conjugated protein and the derived protein. Second type of classification is also there of the protein, and which is also important. And the second type of classification is based on the molecular shape or structure. So the shape that is the based on the classification based on this are divided into two different types, and depending on their molecular shape. So depending upon the molecular shape, the proteins are classified into two types. Number one is a fibrous protein, which are fiber-like, and second is a globular protein, which are globe-like, that is spherical. So again, now this is the second classification of the protein, fibrous protein and globular protein. So first is a fibrous protein. Now what is fibrous protein? It is insoluble in water. In this protein, the polypeptide chain coil about one another and are held together by strong inter-chain hydrogen bonds between hydrogen and oxygen of amino acid unit of the different chain. Therefore, their molecules assume the shape of large linear fibers, which are used for connections, support and the structure in living organism. Examples are here shown: keratin, collagen, myosin, fibrin are the examples of protein that is known as fibrous protein. So that example is the first example, keratin, present in the skin, hair, nail, and second collagen, present in the tendons, cartilages and bones. Third is a myosin, it is present in muscles, and fourth is fibrin, present in the silk. So these are the fibrous protein, which are and also example of this fibrous protein given here. And second is a globular protein, and in this case it is soluble in water or aqueous solution of acid, bases or salt. Their molecules are globular or roughly spherical in shape. They are made up of polypeptide chain coiled back and forth on themselves so as to make a compact spheroid molecules. The polypeptide chain are held together by intra-chain hydrogen bonds between hydrogen and oxygen of amino acid unit of the same chain. The function of globular protein is to maintain and regulate life process in the living organism. And here examples of these globular proteins are given, that is the enzyme, insulin, hemoglobin, antibodies, cytochrome are the examples of this globular protein. As for example, number 1, enzyme. This is the example, and pepsin in stomach and help in digestion. Insulin is the protein. It is secreted by pancreas and regulate glucose metabolism. Hemoglobin, present in the blood, transport oxygen from lungs to all the cellular tissues. Antibodies protect the body from outside the infection. For example, gamma globulin in blood. Cytochrome present in the blood and act as an electron carrier. So these are the functions of this and example and their functions of this globular protein. So these are the two types of classification. Depending upon the molecular shape and chemical composition, we can classify protein into different types. And after this we are going to see the structure of protein. Now depending upon the structure of protein are quite complex, and their structures are generally discussed at 4 different levels, that is primary, then secondary, then tertiary and quaternary structures. So different structures are there, and we can 4 are there, but here we are discussing only the primary and secondary, which are more important. And so first structure is the primary structure of protein, and in this case primary structure, protein may have one or more polypeptide chain. The primary structure of protein refers to the covalent structure including disulfide bridges of each polypeptide chain. Simply, it refers to the sequence in which the various amino acids present in the protein part are linked to one another, and it is shown diagrammatically also. And protein are linked one by one. Here is shown. This is the primary structure of protein, and in this way, and this contain peptide linkage CONH. So this is the CONH group present here, CONH group. So one by one this amino acid group join together by peptide, and that is also known as sometimes polypeptides, and so they contain peptide linkage. And here this primary structure of protein or polypeptide can be determined by findings answer to the and can be determined, and the 2 important factors are here, and that is structure can be determined which involve the 2 steps, that is the determination of amino acid composition, and second step is the sequencing of amino acid. So sequencing of amino acid can be known by the thermal residue analysis method and also N-terminal residue analysis. So C-terminal, N-terminal residue analysis are there. So by using this we know the structure, primary structure of this protein. So determination of amino acid composition and sequencing of amino acid, and sequencing is obtained by C-terminal and N-terminal residue analysis. So this is the first structure, also known as only contain amino acid and also known as polypeptide. So amino acid joined one by one, and there is they link together by the peptide linkage. So also known as polypeptides. The compound is also known as polypeptide, and so this is the primary structure. Primary structure is important, and now second is number 2 is the secondary structure of the protein. And as we know that the conformation which the polypeptide chain assume as a result of hydrogen bonding is known as the secondary structure of the protein. And depending upon the size of the R alkyl group, the following two different structures are possible. One is the alpha helix structure, and second is the beta plated structure, sheet structure. That is, so alpha helix structure and beta plated sheet structure. 2 structures are possible. And what is now the alpha helix structure, which is known as secondary structure of protein. So this is the diagram shows alpha helix structure of the compound, and in this case alpha helix model was given by Linus Pauling, and purely on the theoretical ground, and it is also verified experimentally. If the size of the alkyl group is quite large, the hydrogen bond are formed between the CO of one amino acid residue and the NH of the 4th amino acid residue in the chain. This causes the polypeptide chain to coil up into the spiral structure, and which is known as right-handed alpha helix structure, which is shown here, right-handed alpha helix structure. So this is shown right-handed. So this is the structure. See here now, in this way, this is the right-handed structure, and here is also shown the groups. And in this structure, alpha helix is also known as helix because since each turn of the helix has approximately 3.6 amino acids and 13 membered ring, each formed by hydrogen bonding, and the distance between the two successive terms is a 5.4 angstrom unit. So this is the primary structure, and here this is the alpha helix structure. The alpha helix may be right-handed or left-handed. All the alpha amino acid except glycine are optically active and all have L configuration. And here the helical structure of protein is always right-handed, that is, polypeptide chain turns in the clockwise direction along the helical axis, just as thread of a screw or bolt are right-handed like that. Therefore, stable helix structure result only if it is right-handed. So it is stable when it is right-handed, and they show only the right-handed structure. So many fibrous proteins such as alpha keratin in hair, nail, skin, bead, clots, myosin in muscles have alpha helix structure through the globular protein also contains segment. So this is the first structure, which is known as alpha helix structure, and second is this is the alpha helix structure. Details about this. So this model was given by Linus Pauling and also verified experimentally, and size depends upon the alkyl group, quite large. And second is the beta pleated sheet structure. So here diagram is also shown, and this structure was also given by Linus Pauling, and the simplest possible arrangement of the peptide chain is the one in which the peptide chain are fully extended to form a flat zigzag structure. So see the extended peptide chain structure shown here in this case. So this is the extended peptide chain. So here CONH, here CONH, so but this is extended, and here is the sheet like structure. So this is the one layer, this is another layer, third layer like that, and shown the distance also here. So see the same, this is one chain, two chains are joined, and here is a CO group of the one molecule, NH of the other, and this dash is nothing but the hydrogen bond, intermolecular hydrogen bond. So see here, this is the dash is the inter, and because of this intermolecular hydrogen bonding, we get a sheet structure in this way, one by one, the side by side to form the flat sheet like structure. So they have zigzag structure, or the chain may lie side by side to form the flat sheet structure. So this is the each chain is held hydrogen bond to the two neighboring chain. So this is the hydrogen bond shown in the middle of the two chain. So this is one chain, this is second chain, and hydrogen bonds are shown, and they join the two chain by hydrogen bonding. So this is the beta pleated sheet structure, and here so we can here that we have seen the two types of structure, primary structure and the secondary structure of the protein. And one important part in this case is the denaturation of the protein. So what do you mean by denaturation of the protein? So here protein denaturation means that protein are very sensitive to the action of heat, mineral acid, alkali. On heating or on treatment with the mineral acid, soluble forms of protein such as globular protein often undergo coagulation or precipitation to form fibrous protein, which are insoluble in water. This coagulation also results in the loss of the biological activity of the protein, and that is why the coagulated proteins so formed are known as denatured protein. So again I am repeating that the coagulation also results in the loss of biological activity of the protein, and that is why the coagulated proteins so formed are called denatured protein. Chemically, denaturation does not change the primary structure but bring about the changes in the secondary and tertiary structure of the protein. And the most common example of denaturation of protein is the coagulation of albumin present in the white of an egg. When the egg is boiled hard, the soluble globular protein present in the egg is denatured, resulting in the formation of insoluble fibrous protein. And another example of denaturation is the coagulation that occur when milk is heated with acid, that is like a lemon juice or tartaric acid, then it forms the cheese. During the denaturation, the globular milk protein lactoalbumin become fibrous. So we can say that it was shown that denaturation was irreversible. However, it has been shown that in some cases the process is actually reversible, and if the process is reversed, then it is known as renaturation. Generally it is irreversible, but some cases it get reversed, and so this reverse process is known as renaturation. So we are here important is the denaturation, and which is shown diagrammatically here. What is denaturation? So this is the native protein actually, and denatured protein. So shown in this way. So random, and this is the globular protein, like in the egg, undergo action of heat and then show this type of behavior. So this is the native protein, and after denaturation, the denatured protein shown in this way, random coil type. So native protein and this denatured proteins are different, and depending upon the action of, so there is these proteins are very sensitive to the action of heat, mineral, acid and alkalis. So this is known as denatured protein, and so we have seen today the different aspect about the protein, what is protein, and after that the classification of protein depending upon the chemical molecular shape and the chemical composition. Different types of proteins are there, and also we have discussed the two important structure, primary and secondary structure of the protein, and lastly the what is meant by denaturation of the protein. So these are the different aspects about the protein that we have discussed in today's presentation.